Welcome back to Studious. Today, we are taking a microscopic journey into the hidden factories of your body. We are opening up a visual atlas of intraepithelial glands. In this comprehensive guide, we will be exploring their form, their function, and the incredible secretory pathways that keep you alive. Think about it. Every time you sweat during a workout, digest a delicious meal, or even just blink your eyes, you rely entirely on these tiny, specialized glandular structures. Without them, our internal environment would completely dry up and shut down. But how exactly do these microscopic machines work? How do they know what to release and when? Grab your favorite study snacks, hit that subscribe button, and let's zoom in to uncover the secrets of the intraepithelial glands.
So, what exactly are we looking at? By definition, these glands are specialized tissue groups that are highly organized to secrete substances into the body for either use or discharge. When we look at their architecture, they are primarily constructed from epithelial cells that are either cube-shaped, known as cuboidal, or column-shaped, known as columnar. But here is the major distinction that sets them apart. Intraepithelial glands are essentially exocrine glands that are nested directly within an epithelial layer. A perfect example of this nesting can be found in the posterior lingual glands, which are located right beneath the papillae of your tongue. Instead of releasing hormones into the bloodstream, like endocrine glands, these exocrine wonders push their specialized chemical cocktails directly onto the surface of your skin or the linings of your organs to maintain local moisture and protection.
Our bodies don't just use one method to secrete substances. They have three distinct mechanisms. The first is apocrine secretion, which involves discharging membrane-bound vesicles. You'll find this method heavily utilized in mammary glands and specific types of sweat glands. Next is the merocrine mechanism, which is actually the most common exocrine mechanism in the human body. It involves the careful exocytosis of proteins and glycoproteins, and this is exactly what drives the function of your salivary glands. Finally, we have the most dramatic method, holocrine secretion. In this mechanism, there is a total discharge of the entire secretory cell. The cell literally sacrifices itself to release its contents. This explosive, cell-destroying method is what drives your sebaceous or oil glands.
Now, let's meet the protectors of the microscopic world, goblet cells. These fascinating unicellular glands have one primary function. They secrete mucus to form a vital protective layer over the mucosa of your internal organs. Looking at their architecture, they have a highly distinctive cylindrical or round shape. If you look closely at a histological stain, you'll see they feature a basally located nucleus at the bottom, while the top is packed with apical membrane-bound secretory granules containing all that mucus. Where are they hiding? They are distributed widely across your respiratory tract, including the trachea and bronchi, as well as throughout your digestive system, in the small and large intestines, and even up in the conjunctiva of your upper eyelid to keep your eyes from drying out.
Moving to the surface of our bodies, we encounter the skin defenders, the sebaceous glands. Remember that explosive holocrine mechanism we just talked about? Sebaceous glands use exactly that, functioning as a holocrine-secreting multi-acinar gland, meaning they are composed of multiple secretory portions called acini. The product of all this cellular sacrifice is sebum. This oily, lipid-rich substance is continuously produced to lubricate your skin and coat the shaft of every hair follicle, keeping them soft and preventing water loss. You will find these oily defenders present across almost all areas of human skin, but there are two strict exceptions. You will never find sebaceous glands on the palms of your hands or the soles of your feet, which is why your hands don't get naturally oily.
Next up are the ultimate cooling systems, the thermoregulators, or sweat glands. Their architecture is quite elegant. They are simple tubular structures that feature a coiled secretory base deep in the skin, which leads to a straight excretory duct lined with stratified cuboidal or columnar epithelia. We classify these into two main types. First are the merocrine sweat glands, which are the predominant type and are distributed extensively throughout most areas of the human skin to help cool us down. Then we have the apocrine sweat glands. These are highly localized variants found specifically in the axillary region, your armpits, the areolar and nipple regions, as well as perineal areas and external genitalia. These apocrine glands secrete a thicker fluid that, when broken down by skin bacteria, is the primary cause of body odor.
To really understand these structures, we need to decode their architectural blueprints. Let's look at major salivary glands, which are classified as compound tubular alveolar structures. This sounds complicated, but it just defines their physical shape. Tubular alveolar, or acinar, describes the shape of the terminal secretory units, where the branches finally end. These specific units converge into a fascinating mix of distinct tube-like shapes and sac-like alveolar shapes. But what does the compound part mean? This simply denotes a duct system that branches extensively, much like the limbs of a tree, rather than relying on a single unbranched duct. All of these tiny branches eventually pool their secretions together, sending the fluid out through the main excretory duct.
Within these salivary systems, we have a tale of two cells, serous versus mucus. Serous cells utilize the merocrine secretion mechanism to produce a highly watery, isotonic fluid. This watery fluid is packed with enzymes like amylase to jumpstart digestion. You will find these serous cells primarily located in the parotid gland and the pancreas. On the flip side, we have mucus cells. These also use the merocrine mechanism, but their output is vastly different. Instead of a watery fluid, they produce highly viscous, mucin-containing secretions via their apical granules. This thick mucin is perfect for lubrication and is heavily concentrated in the sublingual gland, the bronchial mucosa in your lungs, and your nasal mucosa.
Nature rarely deals in absolutes, which brings us to the hybrid unit, mixed seromucus glands. As the name suggests, the structure of these glands is designed to produce a hybrid mixture of both serous and mucus secretions, all contained within a single complex. The most defining and frankly beautiful histological feature of this hybrid is known as von Ebner's demilunes. Under a microscope, these appear as crescent moon-shaped serous cells, the demilunes, that physically wrap around and partially surround the central mucus cells. It is a stunning example of cellular cooperation. If you're looking for this specific hybrid architecture in the human body, the primary example is the submandibular gland, located right along your jawline.
Let's officially map out the salivary trio, along with some specialized urethral glands. First is the parotid gland, located bilaterally in the preauricular area, just in front of your ears. It is a purely serous gland and is surprisingly responsible for about 30% of the body's total saliva production. Next is the submandibular gland, which, as we just discussed, is a mixed seromucus gland featuring those beautiful von Ebner's demilunes. Rounding out the trio is the sublingual gland. Located right under the tongue, it functions primarily as a mucus-secreting gland. Beyond the mouth, we also have urethral glands. These are mucus-secreting intraepithelial glands found in both sexes. They are called the glands of Litra in males and Skene's glands in females.
Creating the secretion is only half the battle. Transporting it is the other. Let's trace the duct pathway. It all originates at the intercalated ducts. These are the terminal narrow ducts that connect directly to the secretory unit. They are lined with a simple or low columnar epithelium. To keep the fluid moving, they are tightly wrapped by specialized myoepithelial cells that physically contract to push the secretory product forward. As the fluid travels, it enters the striated ducts, which act as modifiers. These are a direct continuation of the intercalated ducts. Their defining feature is a heavily folded basal membrane. This isn't just for looks, it's a crucial architectural adaptation that facilitates the active transport of water and ions, modifying the fluid before it leaves.
Continuing down our biological highway, the modified fluid finally reaches the interlobular ducts. Connected directly to the striated ducts we just mentioned, these structures act as the body's primary excretory channels. Their structural routing is highly specific. They run explicitly within the connective tissue septa. These septa are essentially structural walls that divide the larger glands into smaller individual lobules. Because the fluid has already been modified and the sole function of these ducts is now just rapid transport to the surface, we see a dramatic cellular shift. Instead of low or folded cells, these interlobular ducts are lined with tall columnar epithelium, highly elongated, column-shaped cells that provide a smooth, fast pathway for final excretion.
Histology isn't just textbook theory. It has real-world clinical applications, like understanding acne vulgaris or common pimples. Remember the sebaceous glands? Their structural holocrine mechanisms are directly linked to the pathology of acne. A healthy follicle allows sebum to flow freely. However, the pathological condition of acne is typically triggered by a massive, sudden increase in the production of that sebum. When does this happen? This overproduction most commonly occurs during the extreme hormonal shifts of adolescence. This massive surge of oil production physically overwhelms the gland's multi-acinar architecture. The result? Severe follicular blockages, creating the perfect environment for bacteria to thrive and painful inflammation to take hold on the skin.
To bring it all together, let's review the intraepithelial synthesis matrix. Goblet cells use a merocrine mechanism to secrete protective mucus in the trachea and intestines. Sebaceous glands use that explosive holocrine method to secrete oily sebum onto the skin, excluding the palms and soles. Sweat glands utilize both merocrine and apocrine methods to release water and lipids across widespread skin and localized axillary areas. The parotid gland is purely merocrine, secreting serous, isotonic fluid near the ear. The submandibular gland is a mixed merocrine gland, secreting a seromucus mixture along the jaw. Finally, the sublingual gland beneath the tongue uses a merocrine method to secrete viscous mucin. Thank you for studying with Studious. Don't forget to like and subscribe.